Hi friends, my name is Nandita. I am from Andhra Pradesh.
Good morning dear students. Today we are going to discuss about photosynthesis in higher plants from Chapter 13 CBSE syllabus plus 1. Children, all animals including human beings depend on plants for their food. Have you ever wondered from where plants get their food? Green plants in fact have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make or rather synthesize the food they need through photosynthesis and are therefore called autotrophs. You have already learned that the autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are heterotrophs. Green plants carry out photosynthesis, physicochemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living organisms on the earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on earth. Photosynthesis is important due to two reasons. It is the primary source of all food on earth. It is responsible for the release of oxygen into atmosphere by green plants. Have you ever thought what would happen if were no oxygen to breathe? This chapter focuses on the structure of the photosynthesis machinery and the various reactions that transform light energy into chemical energy. Children, let us see what do we know. Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, means green pigment of the leaves, light and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation into leaves. Variegated leaf or leaf that was partially covered with black paper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaf in the presence of sunlight. Another experiment you may have carried out where a part of a leaf is enclosed in a test tube containing some KOH cotton which absorbs CO2 while the other half is exposed to air. The setup is then placed in light for some time. On testing for the presence of starch later into two parts of the leaf, you must have found that the exposed part of the leaf tested positive for starch while the portion that was in the tube tested negative. This showed that CO2 was required for photosynthesis. Can you explain how this conclusion could be drawn? Let us see the earlier experiments once again. It is interesting to learn about those simple experiments that lead to a gradual development in our understanding of photosynthesis. Joseph Priestley from 1733 to 1804 in 1770 performed a series of experiments that revealed the essential role of the air in the growth of green plants. Priestley, you may recall, discovery of oxygen in 1774. Priestley observed that a candle burning in a closed place, a bell jar, soon gets extinguished. Similarly, a mouse could soon suffocated in a closed space. He concluded that a burning candle or an animal that breathed the air, both somehow damaged the air. But when he placed a mint plant in the small bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows. Plant restore to the air whatever breathe animals and burning candles remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to Rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight, John Ingenhousz 1970 to 1979 showed that sunlight is essential to the plant process that somehow purifies the air filled by burning candles or breathing animals. Ingenhousz, in an elegant experiment with an aquatic plant, showed that in bright sunlight some bubbles were formed around the green plant while in the dark they did not. Later he identified these bubbles to be of oxygen. Hence, he showed that it is only the green part of the plant that could release oxygen. Was not until about 1854 that Julius I provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. His later studies showed that the green substance in plants, chlorophyll as we know it now, is located in special bodies called chloroplast within the plant cell. He found that the green parts in the plant is where glucose is made and that the glucose is usually stored as starch. Now consider the interesting experiment done by T.W. Engelmann from 1843 to 1909. Using a prism, he split light into its spectral components and then illuminated a green algae Cauliflower placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of photo-evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and B, which is discussed in section 13.4. By the middle of the 19th century, the key features of the plant of photosynthesis were known, namely the plant could be used sunlight energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organisms was then understood as CO2 plus H2O gives rise to CHO2 plus O2. Where CHO2 represented a carbohydrate, means glucose as 6-carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by a microbiologist, Cornelius Neal, from 1897 to 1985, who based on his studies for purple and green bacteria demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. This can be expressed by 2H2A plus 6CO2 gives rise to 2A plus CH2O plus H2O. In green plants, H2O is the hydrogen donor and it is oxidized to O2. Some organisms do not release O2 during photosynthesis. When H2S instead is the hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organism and not O2. Hence, he inferred that the O2 evolved by the green plant comes from H2O, not from carbon dioxide. This was later provided by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore 6CO2 plus 12H2O gives rise to C6H12O6 plus 6H2O plus 6O2. Whereas C6H12O6 represents glucose, the O2 released is from water. This uses radioisotope techniques. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate are used in the equation given above? Now let us discuss where does photosynthesis takes place. Children, you would of course answer in the green leaf or the chloroplast. Based on what you earlier read in the chapter 8, you are definitely right. Photosynthesis does takes place in the green leaves of plants, but it does so also in other green parts of the plants. Can you name some of the other parts where you think photosynthesis may occur? Yes, you would recollect from previous unit that the mesophyll cells in the leaves have a large number of chloroplasts. Usually, the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplasts aligned with their flat surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is a membranous system consisting of grana. The stroma, lamellae, and the matrix stroma are present. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn form starch. The former set of reactions, since they are directly light driven, are called light reactions. The latter are not directly light driven but are depend on the products of light reactions. Hence, to distinguish the latter, they are called by convections as dark reactions. However, this should not be construed to mean that they occur in darkness or they are not light dependent. Children, now let us discuss how many types of pigments are involved in photosynthesis. Looking at a plant, you have ever wondered why and how there are so many shades of greens in their leaves. Even then, in the same plant, we can look for an answer for this question by trying to separate the leaf pigment of any green plant through paper chromatography. A chromatographic separation of a leaf pigment shows that the color that we see in the leaf is not due to a single pigment but due to 4 pigments. One is chlorophyll A, which means bright or blue-green in the chromatogram. Second one is chlorophyll B, yellow-green. Third one is xanthophyll, yellow. And fourth one is carotenoids, yellow or yellow to orange. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelengths. You can guess which is the most abundant plant pigment in the world. Let us study the graph showing the ability of chlorophyll, a pigment that absorbs light of different wavelengths. Of course, you are familiar with the wavelength of the visible spectrum of light as well as VBR. You can determine the wavelength means color of light at which chlorophyll shows a maximum absorption. Does it show another absorption peak at any other wavelength too? If yes, which one? Now look at the figure showing the wavelength at which the maximum photosynthesis occurs in a plant. You can see that the wavelength at maximum absorption by chlorophyll, that is in the blue and the red regions, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at the figure, can you say that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action of spectrum of photosynthesis? Children, these graphs together show the most of the photosynthesis takes place in blue and red regions of the spectrum. Some photosynthesis does not take place at the other wavelength of the visible spectrum. Let us see how this happens through the chlorophyll is the major pigment responsible for trapping light. Other picoid pigments like chlorophyll B, xanthophylls and carotenoids, which are also called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they do not only enable a wide range of wavelength of incoming light to be utilized for photosynthesis, but also protect chlorophyll, a form of photooxidation. Children, let us see what is light reaction. Light reaction or light or photochemical phase includes light absorption, water splitting, oxygen release, and the transformation of high energy chemical intermediates ATP and NADPH. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes like LHC within the photosystem PSI and photosystem 2 PS2. Photosystem 1 PS1 and photosystem 2 PS2. They are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LHC are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigments forming a light harvesting system also called antennae. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll, a molecule, forms the reaction center. The reaction center is different in both the photosystems. In PS1, the reaction center chlorophyll A has an absorption peak at 70 nm, hence it is called P700, while in PS2, it has absorption maximum at 680 nm and it is called P680. Children, let us discuss the electron transport. In photosystem 2, the reaction center chlorophyll A absorbs 680 nm wavelength of red light, causing electrons to become excited and jump into an orbit further from the atomic nucleus. These electrons are picked up by an electron expector which phases them to an electron transport system consisting of cytochromes. This movement of electrons is downhill in terms of an oxidation reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain but are passed on to the pigments of photosystem PS1. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nm and are transferred to another aspector molecule that has a greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy rich NADPS+. The addition of these electrons reduces NADP+ to NADPH+ H+. This whole scheme of transfer of electrons starting from the PS2 uphill to the aspector down the electron transport chain to PS1, excitation of electrons, transfer to another aspector and finally downhill to NADP+, reducing it to NADP plus H plus is called the Z scheme. Due to its characteristic shape, the shape is formed when all the carriers are placed in a sequence on a redox potential scale. Children, now let us discuss about splitting of water. You would then ask, how does PS2 supplies electrons continuously? The electrons that we were moved from photosystem 2 must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the PS2. Water is split into 2H plus O and electrons. This creates oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from photosystem 1 and provided by photosystem 2. Children, we need to emphasize here that the water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of the thylakoid. Then, where are the protons and SO2 formed likely to be released in the lumen or on the outer side of the membrane? Children, now let us discuss about cyclic and non-cyclic photophosphorylation. Children, living organisms have the capability of extracting energy from oxidizable substance and store this in the form of bound energy. Special substances like ATP carry this energy in their chemical bonds. The process through which the ATP is synthesized by cells is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first PS2 and then the first PS1, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain as seen earlier in Z scheme. Both ATP and NADP plus H plus are synthesized by this kind of electrons flow. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons. A possible location where this could be happening is in the stroma lamella. While the membrane of lamellae of the grana have both PS1 and PS2, the stroma lamellae membrane lack PS2 as well as NADP reductase enzyme. The excited electron does not pass on to NADP but it cyclized back to the PS1 complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not of NADP H+. Cyclic phosphorylation also occurs when only light of wavelengths beyond 680 mm are available for excitation. Children, let us discuss about chemoosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemoosmotic hypothesis has been put forward to explain the mechanism like in respiration, in photosynthesis too, ATP synthesis is linked to development of a proton gradient across a membrane. This time these are the membranes of thylakoids. There is no one difference. Here the proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration, protons accumulate in the intermembrane space of the mitochondria where electrons move through the ETS. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that take place during the activation of electrons and their transport to determine the steps that causes a proton gradient. Since splitting of water molecules takes place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. As electrons move through the photosystem, protons are transported across the membrane. This happens because the primary aspector of electrons, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an H carrier. Hence, this molecule a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. Then NADP reductase enzyme is located on the stroma side of the membrane. Along with electrons that come from the aspector of electrons, PS1 protons are necessary for the reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decreases in the number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as measurable decrease in pH in lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the CFO and the ATP synthase. The ATP synthase enzyme consists of two parts. One called that CF0 is embedded in the thylakoid membrane and forms the transmembrane channel. The gradient is broken down due to the movement of proton across the membrane up to the stroma through the transmembrane channel of the CF0 of ATP synthase. The ATP synthase enzyme consists of two parts. One is called CF0 is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of ATP synthase, which makes the enzyme synthesize several molecules of energy-packed ATP. Chemoosmosis requires a membrane, a proton pump or a proton gradient and ATP synthesis, etc. Energy is used to pump protons across a membrane to create a gradient or a high concentration of portions within the thylakoid lumen. Its synthase is a channel that allows diffusion of the protons back across the membrane. This releases enough energy to active ATP synthase enzymes that catalyzes the formation of ATP. Along with NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reactions takes place in the stroma, responsible for fixing CO2 and synthesis of sugars. Children, let us discuss where are the ATP and NADP used. We learned that the products of light reactions are ATP, NADPH and O2. Of this, O2 diffuses out of the chloroplast while ATP and NADPH are used to drive the process leading to the synthesis of food, more accurately sugars. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light, but it is depend on the products of the light reaction, that is ATP and NADPH besides CO2 and H2O. You may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, this biosynthetic process continues for some time and then stops. If then the light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as the dark reaction is misnomer? Discuss this among us yourself. Let us now see how the ATP and NADP are used in the biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce 6H2O or sugar. It was of interest to scientists to find out how this reaction proceeded, or rather that what was the first product formed when CO2 is taken into a reaction or fixed. Just after World War II, among the several efforts to put radioisotopes to beneficial use, the works of Melvin Calvin is exemplary. The use of radioactive 14C by him is a algal photosynthesis studies lead to the discovery that the first CO2 fixation product was a 3-carbon organic acid. He also contributed to working out the complete biosynthetic pathway. Hence, it is called Calvin cycle after him. This first product identified was 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also try to know whether all plants have PGA as the first product of CO2 fixation, or whether any other product was formed in other plants. Experiments conducted over a wide range of plants lead to the discovery of another group of plants, where the first stable product of CO2 fixation was again an organic acid, but one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid, or OAA. Since then, CO2 assimilation during photosynthesis was said to be of two main types: Those plants in which the first product of CO2 fixation is Ca3 acid PGA, that is the C3 pathway, and those in which the first product was C4 acid and OAA, that is the CO4 pathway. There are two groups of plants showed other associated characteristics that we will discuss later. Children, now let us discuss about the primary acceptor of CO2. Let us now ask ourselves a question that we asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would be a molecule have which after accepting fixing CO2 would have 3 carbons of PGA? The study very unexpectedly showed that the acceptor molecule was a 5 carbon ketose sugar ribulose biphosphate RUBP. Did any one of you think of this possibility? Do not worry, the scientists also looked a long time and conducted many experiments to reach this conclusion. They also believed that since the first production was a C3 acid, the primary acceptor would be a 2 carbon compound. They spent many years trying to identify a 2 carbon compound before they discovered the 5 carbon RUBP. Children, now let us discuss about the Calvin cycle. Calvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Calvin pathway operates and where the sugar is synthesized. Let us at the outside understand very clearly that the Calvin pathways occurs in all photosynthetic plants. It does not matter whether they have C3 or C4. For ease of understanding, the Calvin cycle can be described under 3 stages, that is carboxylation, reduction and regeneration. The carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of Calvin cycle where CO2 is utilized for carboxylation of RUBP. This reaction is catalyzed by the enzymes RUBP carboxylase, which results in the formation of 2 molecules of 3-PGA. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase oxygenase or RUBIS-O2. Discuss about reduction. There are a series of actions that lead to the formation of glucose. The steps involve utilization of 2 molecules of ATP for phosphorylation and 2 of NADP for reduction per CO2 molecule fixed. The fixation of 6 molecules of CO2 and 6 turns of cycle are required for the formation of 1 molecule of glucose from the pathway. Now let us see what is regeneration. Regeneration of CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP. Hence, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 of NADP are required. It is probably to meet this difference in number of ATP and NADP used in the dark reactions that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of cycle are required. Work out how many ATP and NADP molecules will be required to make 1 molecule of glucose through the Calvin pathway. It might help you to understand all of this if you look at what goes in and what comes out of the Calvin cycle. Children, now let us discuss about C4 pathway. Plants that are adapted to dry tropical regions have the C4 pathway mentioned earlier. Though these plants have the C4 oxaloacetic acid as the first CO2 fixation product, they use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then, in what way are they different from C3 plants? This is a question that you may also reasonably ask. C4 plants are special. They have a special type of leaf anatomy. They tolerate higher temperatures. They show a response to high light intensities. They lack of process called photorespiration and have a greater productivity of biomass. Let us understand this one by one. Study vertical sections of leaf on one of a C3 plant and other of a C4 plant. Do you notice the difference? Do both have the same type of mesophils? Do they have similar cells around the vascular bundle sheath? The particularly large cells around the vascular bundle of C4 plants are called bundle sheath, and their leaves which have such anatomy are said to have Kranz anatomy, and Kranz means width and is a reflection of the arrangement of leaves. The bundle sheath cells may form several layers around the vascular bundles. They are characterized by having a large number of chloroplasts, thick walls, impervious to gaseous exchange, and no intracellular spaces. You may like to cut a section of the leaf of C4 plants, maize or sorghum, to observe the Kranz anatomy and the distribution of mesophyll cells. It would be interesting for you to collect leaves of diverse species of plants around you and cut vertical sections of the leaves. Observe under the microscope, look for the bundle sheath around the vascular bundles. The presence of the bundle sheath would help you to identify the C4 plants. Now study the pathway shown in the figure. Pathway that has been named the Hatch and Slack pathway is again a cyclic process. Let us study the pathway by listing the steps. The primary CO2 acceptor is a 3-carbon molecule, phosphophenol pyruvate, and it is present in the mesophyll cells. The enzyme responsible for this fixation is PEP carboxylase or PEP case. It is important to register that the mesophyll cells lack RUBS coenzyme. The C4 acid OAA is formed in the mesophyll cells. It then forms other 4-carbon compounds like malic acid and aspartic acid in the mesophyll cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release CO2 and 3-carbon molecules. The 3-carbon molecules is transported back to the mesophyll where it is converted to PEP again, thus completing the cycle. The CO2 released in the bundle sheath cells enter the C3 or the Calvin pathway, and a pathway common to all the plants. The bundle sheath cells are rich in an enzyme ribulose biphosphate carboxylase oxygenase, but lack in PEP case. Thus, the basic pathway that result in the formation of sugars, the Calvin pathway, is common to the C3 and C4 plants. Did you notice that the Calvin pathway occurs in all the mesophyll cells of the C3 plants? In the C4 plants, it does not take place in the mesophyll cells but does so only in the bundle sheath cells. Children, now let us discuss about photorespiration. Let us try and understand one more process that creates an important difference between C3 and C4 plants, photorespiration. To understand photorespiration, we have to know a little bit more about the first step of Calvin pathway, the first CO2 fixation step. The reaction where RUBP combines with CO2 to form two molecules of 3-PGA that is catalyzed by RUBis CO and RUBP-CO PGA. RUBP-CO, that is the most abundant enzyme in the world, and it is characterized by the fact that its active site can be bent to both CO2 and O2, hence the name. Can you think how this could be possible? RUBP-CO has much greater affinity for CO2 when the CO2 and O2 is nearly equal. Imagine what would happen if this were not so. This binding is competitive. It is the relative concentration of O2 and CO2 that determines which one of the two will bind to the coenzyme. In C3 plants, some O2 does bind to RuBisCO and hence O2 fixation is decreased. Here the RuBP, instead of being converted to two molecules of PGA, binds with O2 to form one molecule of phosphoglycerate and phosphocholate 2 carbon in a pathway called photorespiration. In the photorespiration pathway, there is neither synthesis of sugar nor of ATP. Rather, it results in the release of CO2 with the utilization of ATP. In the photoreceptor pathway, there is no synthesis of ATP or NADP. The biological function of photorespiration is not known yet. In CO4 plants, photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. This takes place when the C4 acid from the mesophyll is broken down in the bundle sheath shells to release CO2. This results in increasing the intracellular concentration of CO2. In turn, this ensures that the RuBisCO functions as a carboxylase, minimizing the oxygenase activity. Now that you know that the C4 plants lack photorespiration, you probably can understand why productivity and fields are better in these plants. In addition, these plants show tolerance to higher temperature. Based on the above discussion, you can compare plants showing the C3 and C4 pathways. Children, now let us discuss about factors affecting photosynthesis. An understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the field of plants, including crops. Photosynthesis is understand the influence of several factors, both internal and external of the plant. The plant factors include the number, size, age, and orientation of leaves, mesophyll cells, and chloroplast, internal CO2 concentration, and the amount of chlorophyll. The plant or internal factors are dependent on the genetic predisposition and the growth of the plant. The external factors would include the availability of sunlight, temperature, CO2 concentration, and water. As a plant photosynthesis, all these factors will simultaneously affect its rate. Hence, through several factors interact and simultaneously affect photosynthesis or CO2 fixation. Usually, one factor is the major cause or is one that limits the rate. Hence, at any point, the rate will be determined by the factor available at suboptimal levels. When several factors affect any biochemical process, Blackman's 1905 law of limiting factors comes into effect. This states the followings: if a chemical process is affected by more than one factor, then its rates will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. For example, despite the presence of green leaf and optimal light and CO2 conditions, the plant may not photosynthesize if the temperature is very low. This leaf is given the optimal temperature will start photosynthesizing. Children, now let us discuss about light. We need to distinguish different light quality, light intensity, and the duration to explore two light. While discussing light as a factor that affects photosynthesis, there is a linear relationship between incident light and CO2 fixation rates at low intensities. At higher light intensity sites, gradually does not show further increase as other factors become limiting. What is interesting to note is light saturation occurs at 10% of the full sunlight. Hence, except for plants in shade or in dense forest, light is rarely a limit factor in nature. Increase in incident light beyond a point cause the breakdown of chlorophyll and a decrease in photosynthesis. Children, now let us discuss about carbon dioxide concentration. Carbon dioxide is the major limiting factor for photosynthesis. The concentration of CO2 is very low at the atmosphere, means between 0.03 and 0.04%. Increase in C concentration up to 0.5% can use an increase in CO2 fixation rates. Beyond this, the level can become damaging over longer periods. The C3 and C4 plants respond directly to CO2 concentration. At low light condition, neither group responds to high CO2 condition. At high light intensities, both C3 and C4 plants show increase in the rate of photosynthesis. What is important to note that the C4 plants show saturation about 360 milliliters, while C3 responds to increased CO2 concentration and saturation is seen only beyond 450 milliliters. Thus, current availability of CO2 levels is limiting to the C3 plants. The fact that C3 plants respond to higher CO2 concentration by showing increased rate of photosynthesis leading to higher productivity has been used for some greenhouse crops such as tomatoes and bell pepper. They are allowed to grow in a carbon dioxide enriched atmosphere that leads to higher yields. Children, now let us discuss about temperature. The dark reactions being enzymatic are temperature controlled. Though the light reactions are also temperature synthesized, they are affected to a much lesser extent. The C4 plants respond to higher temperatures and show higher rate of photosynthesis, while C3 plants have a much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depend on the habitat that they are adapted to. Tropical plants have a higher temperature optimum than the plants adapted to the temperature climates. Children, now let us discuss about water. Even though water is one of the reactant in which the light reaction, the effects of water as a factor is more through its effect on the plant rather than directly on photosynthesis. Water stress causes the stomata to close, hence reducing the CO2 availability. Besides, water stress also makes leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well. Green plants make their own food by the process of photosynthesis. During this process, carbon dioxide from the atmosphere is taken by green leaves through the stomata and it is used for making carbohydrates, principally glucose and starch. Photosynthesis takes place only in green part of the plant, mainly in the leaves. Within the leaves, the mesophyll cells have large number of chloroplasts that are responsible for CO2 fixation. Within the chloroplast, the membrane nests are sites for the light reaction. While the chemosynthetic pathway occurs in the stroma, photosynthesis has two stages: the light reaction and the carbon fixation reactions. In the light reaction, the light energy is absorbed by the pigments present in the antenna and funneled to special chlorophyll, a molecule called reaction center chlorophylls. There are two photosystems, one is PS1 and PS2. PS1 has 700 nm absorbing chlorophyll, a P700 molecule at its reaction center, while PS2 has P680 reaction center that absorbs red light at 680 nm. After absorbing light, electrons are excited and transferred through PS2 and PS1 and finally to NAD forming NADH. During this process, a proton gradient is created across the membrane of the colloid. The breakdown of the protons gradient due to the movement through the FO part of ATPase enzyme releases enough energy for the synthesis of ATP. Splitting of water molecules is associated with PS2, resulting in the increase of O2. Protons and transfer of electrons of PS2 in the carbon fixation cycle. CO2 is added by enzyme. RUBP is CO2 5 carbon compound RUBP that is converted to 2 molecules of 3 carbon PGE. This is then converted to sugar by Calvin cycle and the RUBP is regenerated. During this process, ATP and NADP synthesized wastefully synthesized in the light reaction are utilized. RU bis CO are also catalyze a wasteful oxygenation reaction in C3 plants. Photorespiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In these plants, the first product of CO2 fixation that takes place in mesophyll is 4 carbon compound. In the bundle sheet shells, the Calvin pathways is carried out for the synthesis of carbohydrates. Thank you.